Dying epithelial cells must be extruded from the tissue without leaving any gaps that would compromise the epithelium's barrier function. Adjacent healthy epithelial cells form a basal actomyosin ring around their dying neighbour to push the apoptotic cell out of the epithelial layer. But, while he was a postdoc in Luca Primo's lab at the Candiolo Cancer Institute in Italy, Paolo Gagliardi noticed that actomyosin rearrangements in the apoptotic cell itself might also play a role in epithelial extrusion.
We can clearly see that there is complex actin cytoskeleton dynamics within the apoptotic cells. We can see the formation of this ring-like structure in the apical part with the diameter around 2 microns and with the central part in which there is less polymerized actin. And importantly, this ring is associated to multiple actin bundles that converge into it. And it is a highly dynamic structure in which we can clearly see a flow of actin coming from the outside into the ring itself.
This apical actin ring forms before the neighbouring cells assemble a ring around the base of the apoptotic cell. Epithelial extrusion depends on myosin contractility, and it is known that, during apoptosis, caspases can cleave and activate the Rho kinase ROCK1, allowing it to phosphorylate the myosin light chain MLC2 and stimulate myosin activity. Gagliardi and colleagues investigated the role of 3 Rho kinase family members, ROCK1, MRCK alpha and MRCK beta, that appear to contain caspase cleavage sites and are expressed in the epithelial cell line MCF10A.
And we found that only the silencing of MRCK alpha is able to hamper the epithelial extrusion promoted by growth factor deprivation. And the silencing of the other 2 proteins had substantially no effect. So we concluded that MRCK alpha in this model is the main Rho kinase regulating the process of extrusion. In other cell model, we found that also ROCK1 has a prominent role in cell extrusion.
Gagliardi et al. Confirmed that, in MCF10A cells, MRCK alpha is cleaved by caspases during apoptosis, generating an N-terminal fragment with increased kinase activity that could play a crucial role in forming the apical actin ring observed in dying cells.
We expressed the N-terminal part of MRCK alpha in our cell by transfection, and as soon as cell start to express this protein, the same mechanical program that we observed in apoptotic cell is executed. We saw the formation of the apical actin ring associated with the multiple bundles pulling toward the ring, and then after the ring is assembled, we observed also the process of epithelial extrusion. So in sum, the simple expression of the cleavage form of MRCK alpha without the whole program of apoptosis is sufficient to induce epithelial extrusion.
The myosin inhibitor blebistatin prevented the cleaved form of MRCK alpha from inducing apical ring assembly and cell extrusion. Moreover, if the researchers added blebistatin after the ring had already formed, it inhibited the centripetal flow of actin towards the ring, indicating that myosin activity is required for both ring assembly and function. However, unlike in apoptotic cells, where extrusion occurs immediately after apical ring assembly, extrusion is delayed for a period of several hours in cells expressing the N-terminal domain of MRCK alpha.
And we found that this period that we call the tug-of-war phase is due to the fact that the flow of actin in cables toward the ring is compensated by actin polymerization at the end of the actin bundles. And the addition of an inhibitor of actin polymerization, like latrunculin A or cytokalasin, during this tug-of-war phase, causes a rapid contraction of the cell body. And this contraction is due to the myosin contraction, because if we add blebistatin together with an inhibitor of actin polymerization, we completely inhibit the collapse of the cell.
Thus, to drive cell collapse and detachment, the activation of myosin by the cleavage product of MRCK alpha must be coupled with a downregulation of actin polymerization activity at the ends of actin bundles, which are located at cellular adhesion sites. So,
apoptotic epithelial extrusion is a process that depends on both the apoptotic cell itself and the neighboring cell. And the initiating part of extrusion is actually dependent only on apoptotic cell, because it assembles this ring, which is needed to pull upwards the cell body, and then the neighboring cell finish the job by closing the ring on the basal side of the epithelium.
Gagliardi thinks that it will be important to examine the mechanics of the apical actin ring and determine how they differ from those of similar structures, such as a cytokinetic ring that drives cell division. For now, though, you can learn more about how MRCK alpha is activated by caspase cleavage to assemble an apical actin ring for epithelial cell extrusion in the paper by Gagliardi et al., published in the January 2nd, 2018 issue of the Journal of Cell Biology.